trying to.
Sean Cox from Simon Fraser University. As a fisheries scientist, I look at the trends you were showing in productivity and indicators of marine survival since the 1980s, and really the trend in declining survival has been going on for almost 20 years. So I'm wondering how much you care to speculate on, whether what you're seeing now could be responsible for a trend like that versus more extreme events like we saw in 2009.
What we're seeing now in which context? I'm sorry, I'm not sure I understand the question.
Well, what you saw in 2009 in terms of
whether that was a one-off sort of
extreme
situation
mortality
versus part of the same phenomenon.
or whether they're related.
I think that could be quite debated. And the really unfortunate thing about 2009 was that, that wasn't a year that had a lot of people studying sockeye salmon. So there really was very little information about sockeye salmon smolts leaving the Fraser River that year. And sort of by the time we realized that so few fish were coming back, it was almost too late to do the sampling. Our genomics program started in 2008. And we were very, very fortunate in having a small number of samples collected in 2007 so we utilized to the max. But we would have liked much bigger sample sizes than we had. I don't know that we're ever really going to be able to answer that, given the lack of data that were available for those smolts coming out in 2007. I think it certainly is plausible that it could be something, within the scheme of the same phenomena going on. But given the lack of data, I'm not positive that one could really answer that definitively. We certainly have some interesting data to suggest that there was a higher degree of stress and pathogens and some other things going on in that year. And there was some indication of impacts of harmful algal blooms, but whether or not that was highly unusual on the context of 25 years is something that one really has to consider. In the genomics data, we have 4 years and 5 years of data on smolts, more years on adults. So it's hard to answer. I'm not sure I can definitively say.
Maybe I just add, I think the opportunity to start sorting some of those things out you're referring to, Sean, is really coming to life now. The department, I understand, is implementing new downstream programs. Kristi probably knows more than I do right now. But the real value of this, and I was sitting here listening to some of the earlier talks thinking that the fundamental difference between the genomics work with people or mice is they don't disappear. Right? But everything that we do disappears in front of us because we get about 1 to 3% survival coming back. So one of the real values of Kristi and Scott Hinch at UBC and others that participated, Dave Patterson, they really have used all sorts of other technologies so you can really make the most of the information that's being collected by Kristi's genomics research. A really good example now is for the past 2 years we've actually tagged smolts leaving Choco Lake, one of the lakes that Kristi has been monitoring. And so we're putting these active sonic tags in small smolts leaving the lake and looking at their survivorship going down the river out to the Strait of Georgia and to the top of Johnson Strait. No one has ever measured the survivorship of smolts in the river. Well, it turns out that about half the mortality occurs in the river. Right? But what's really interesting is that the smolts stayed in the southern part of the Strait of Georgia for the longest period of time, up to about 25 days, and they suffer about another 50% mortality by the time they get to halfway up Georgia Strait. And we know this because there are active arrays in the bottom of the strait getting the reception of the data, and then the next one is at the top of Johnson Strait. So by actually building on all these tools together, we really are starting to make some progress in understanding where marine survival occurs. We still don't know what, and that's the fascinating part of Kristi's work. And it sort of leads to my question in the sense, one of the things I think will come out, and you're hearing it in the paper already from the Cullen Commission, is a broad-scale sampling of BC's wild salmon to look at the pathogens in the fish. I'm not sure I understand the new methodology we're describing yet, but do we have enough background that we could really do a snapshot of people collected samples throughout BC at one time and really collected presence?
You know, I think that's something that we're building in our lab right now. I think I worry about the approach of a very expensive, broad-scale pathogen screening that only focuses on reportable diseases, which is what's currently being proposed by CFIA and DFO, because that's three viruses that they're going to look at, and there's a plethora of other pathogens, and we've screened those 3 viruses and a plethora of other pathogens, and we know those aren't the ones that are really the most active in wild fish in any of our screening. So I do believe with the technology that we have now, we will be able to look at thousands and thousands of individuals and on the order of 50 or more pathogens that could be monitored simultaneously. And we can, in our lab right now, we can screen 300 or 400 fish for 50 pathogens at once in duplicates. So that technology is there, and that's going to really add a lot of information on which pathogens should we be focusing our attention on, which ones are highly prevalent, which ones show high levels of intensity of infection at the stages in the life history that we believe salmon are undergoing the highest levels of mortality, what's the differential between years where we have different levels of productivity and ocean survival, and really start getting a handle on where we should be putting our laboratory studies and follow-up studies. And I think that the added component of having the genomics data that we already have collected in this program is huge. We have run over 3,000 arrays in our program, comprised of multiple different tissues, and if we run pathogen screens on all of those fish, we can really get a handle on which ones of those pathogens are causing the most damage in those fish at the time of sampling.
Just comment on your point about doing the 3 from CFIA. I think that would be a classic mistake. You hear people now saying that we don't know what's in wild salmon, the pathogens, but the department used to do very, very extensive screening throughout British Columbia, and it was in preparation for the saltwater enhancement program. There was a staff at the biological station that really had some of the top fisheries disease people in the world. So to say that we don't know what's there is really, we don't know necessarily from last year, but you definitely would not want to limit it, because I think what we're really seeing in some of your work is we're detecting things that have probably been there for a while, and we just didn't know to look. And some of this is really a reflection of the tools that are now available to us.
I think that's exactly the case. Yeah.
Any other questions?
It was also came out that nowadays there's a very high percentage of the fish in the North Pacific are hatchery raised, not just from BC but from Alaska, Canada. And is anyone looking at the genomics of hatchery raised fish? And I'm particularly interested because in human beings the gut microbiome is formed in almost hours after birth, and it's quite possible the gut microbiome in hatchery raised fish and wild fish may be significantly different. But is anyone starting to do any systematic work on hatchery raised fish? Because the presence of them was also raised as a potential concern of the common environment.
We actually have been doing that. I showed one slide or so that included some of the hatchery data, but we have performed our microarray experiments on sockeye, chinook, and coho, and the chinook and coho studies would have included both hatchery and wild fish. And Brad Davis in my group is a postdoc and is analyzing, basically, asking the question: Are hatchery fish, do they continue to be physiologically distinct from wild fish after they are cohabiting a common environment in the ocean, and for how long, and in what ways are they divergent from wild fish if they are divergent? And we have some pretty outstanding data on that, and that shows that even sampled into the fall after they've been in the common ocean environment for over 4 months, we still see strong physiological divergence between hatchery and wild fish, and affecting a variety of physiological processes, and that's something that we're writing up right now. In terms of pathogen screens, again, that's going to be very, very important to understand What are the pathogen profiles in hatchery fish versus wild fish? Are there pathogens that are emanating more from the hatchery environment than they are in a wild environment? And are those eventually picked up by fish in a common environment in the ocean? Those are all questions that we have the samples to address, and we will be following up with.
I can say personally that the change in attitude about in major hatcheries, now not the small community hatcheries, the major hatcheries, probably the single largest change I've seen in my lifetime. When the major hatcheries really sprung up in BC would have been through the 70s and into the early 80s. And you really couldn't criticize them. They were going to solve everything. And reality is they've changed nothing. They probably made things worse, right? But nobody would have said that 15 years ago within the Department of Fisheries and Oceans. And there are people, we have a complete design for looking at testing just what the fitness of hatchery fish is by generating strains within hatcheries. And we simply haven't been able to get anybody to fund it. But the interesting thing is the department will now implement it. So there's no question that there's a realization that what we told people was a known technology and it will double catch and all the rest. It has just been really a significant dollar sink and a failure. Now, in some areas, the hatcheries are quite productive, and this is the real dilemma. Robertson Creek hatchery for Chinook and coho salmon is a major income to Port Alberni. But other West Coast hatcheries in the West Coast have really done nothing for the national stocks and maybe retarding them from recovery. And so it's a huge question. Over 6 billion juveniles released into the North Pacific every single year now. And Russia this year will surpass Japan as the biggest hatchery program in the world. And so it's a big issue.
So, Dr. Miller, I think I saw in the slides that the 2008 smolt group seemed to have less pathogens. Was that from all of the
There
slides?
certainly were certain pathogens that were less prevalent in that.
And so, and you think that perhaps correlates with the record returns from 2010?
We're still running more fish and more tissue, so we haven't run all of those correlations. But there certainly are some differentials of specific pathogens that show a decreased prevalence in 2008 relative to 2007.
Any speculation why they might have less pathogens?
Well, we see differentials in pathogens that are coming from freshwater, like Flavibacter, which was 40% of the fish coming out of the river in 2007 and non-existent in 2008, or the other years that we studied it. That's a common. It's a freshwater bacterial pathogen that causes mortality in the freshwater environment. And then there were other ones that really only showed up in the marine environment that were also differentials. If you're asking where they came from, that's going to take a little bit more study. We do know in all of our sampling, where we sample those fish. Okay, I didn't really go over this very well in my talk, but we go through a sampling trajectory in both adults and smolts, where in smolts we're characterizing fish all the way from their natal rearing areas all the way through to the first 6 to 9 months in the ocean. And there's really huge, extensive sampling program that her husband, Mark Riddell, and Dick Beamish have been involved in a huge way, David Patterson. And so we've collected thousands of smolts from multiple species and multiple stocks in that trajectory from natal rearing area to all the way up to Southeast Alaska in multiple years. And then in the returning adults, we sample them sometimes as far as King Charles Islands all the way back to their natal rearing area. So those are the data with which we can start analyzing the presence of pathogens or various signatures. And so we can ask the question, which of these pathogens are emulating out of the freshwater environment? Where are they picking them up? Are they leaving their natal rearing areas with them? Are they picking them up in the lower river or when they first enter the ocean? Or do we not see those pathogens until they get past the Pelago, for instance? So all of those data are available to us to look at. But I can't say we've done a systematic analysis of that yet because we've just started doing the surveys.